I would like first to thank the organizers for their invitation to address an overview about the role of probiotics in gut pathophysiology. I have no conflict of interest. In order to understand how probiotics may target the gut, we have to keep in mind the concept of microbiota-gut-brain axis. At least this concept consists to consider in periphery the gut containing different entities, such as and different cellular populations, such as microbiota, mucus layer, intestinal epithelial cells, immune cells, mucosal immunity, enteric nervous system, and all these connected to the sympathetic and parasympathetic system. This consortium of entities crosstalk regularly in a bidirectional way, and in basal conditions, in healthy conditions, this crosstalk is harmonious and guarantees homeostasis. In disease, this crosstalk can be a little bit cacophonic, and for instance, under deleterious stimuli as inflammation or antibiotic use or unbalanced diet, we can find some disruption in the integrity of the intestinal barrier function, stimulation of the mucosal immunity and also general immunity, and of course neurochemistry, which is altered in these conditions. Local neurochemistry can also sensitize nerve fibers and propagate these adverse messages to the central nervous system, which also respond. So, in these conditions, we can also, of course, have some alterations in the microbiota, and probiotics can be there in order to a little bit change this cacophonic crosstalk. These alterations under deleterious conditions are traduced by alteration in gut functions, and here in this talk we will focus on alterations on gut function as motility or absorption, water and electrolyte absorption, the intestinal barrier function, the visceral sensitivity, the HPA responsiveness to stress and behaviors. All this can be modified, altered in disease. The role of probiotics can be now a little bit more easy to understand, since these microorganisms can act locally in order to establish a relation with the host through action on the epithelial cells, changes in the morphology and the structure of these cells, recognition of epithelial cell receptors, and can also interact with the microbiota in order to avoid some pathogenic invasion and to balance some dysbiotic conditions. So this is in the periphery, and this concept of the probiotics acting in the periphery through this kind of mechanism has been expanded to the concept of psychobiotics, meaning that this action on periphery can also affect the gut-brain axis and produce effects in the central nervous systems and the brain. So the probiotics can be considered now as actors in the lumen, able to affect locally and beyond the health. So let me now start with some paradigms on role of probiotics in gut functions, and first of all we will speak about gut motility, and we will focus on preclinical data. This talk is not exhaustive. Not all the literature about the effect of probiotics on this function has been reported, but I have made a choice of some important data according to me. This is a recent study on an extract of the Bifidobacterium lactis, HNO19. This is an extract isolated from Bifidobacteria, Bifidobacterium lactis, cultured in anaerobic conditions. And the authors have isolated large intestine, and they measured the contraction amplitudes in different sites of this isolated large intestine, from the proximal to the most distal sites, and they have compared the effects obtained after the administration in the lumen of this probiotic versus a prokinetic precalopride. And they have seen that the contraction amplitude named by the authors as synchronous contractions, these propagated contractions from one side to another, were stimulated by the prokinetic, of course, but also by the probiotic. But this effect was occurred for the contraction amplitude of these propagated contractions after the washout, meaning with a delay after administration of the probiotic. In this study also, they have measured the effect of precalopride and Bifidobacterium on the non-propagated contractions, and they have seen that Bifidobacterium lactis extract inhibits the non-propagated contractions in this isolated large intestine, clearly showing that this extract of this probiotic may have a beneficial effect on gut motility. This study deals with yeasts, which is another category of probiotics, and compares the effect of Saccharomyces boulardii and cerevisiae. And here again, it's a study conducted in vitro, and this is the measure of the propagated contractile clusters velocity in the jejunum of animals submitted to stress or controls. Here we can see that the propagated contractile clusters in the jejunum of stressed animals is decreased, and in contrast, in the colon is increased. And the authors have seen here that this modification in the propagation of contractile clusters was restored in the jejunum and in the colon by both yeasts, showing that here again this category of probiotics can be beneficial for motility travelers. Let's move now to the paradigms concerning the effect of probiotics on the intestinal barrier function. Here is a study coming from our lab showing that this probiotic, Lactobacillus farciminis, was able to to prevent the hyperpermeability induced by stress. And in this case, we have shown that this effect on hyperpermeability, on paracellular hyperpermeability, was due to an effect of the probiotic treatment on the MLCK activation. At least it means that the probiotic inhibits the MLCK activation and leads to the myosin light chain phosphorylation, which means contraction of the cytoskeleton and opening of the tight junctions. This action on the cytoskeleton and on the tight junction was confirmed in the other paradigm using a fermented milk product, showing that the fermented milk product was also able to to prevent the hyperpermeability induced by stress. And this activity of the fermented product was was associated with restoration of the expression of oclidin and JAM-A proteins of the time junction complex, showing clearly that here the restoration of the hyperpermeability, epithelial hyperpermeability, can be mediated by an action of the probiotics on the cytoskeleton and tight junction proteins. Here is another study coming from Canada showing that a probiotic combination prevents also some gut dysfunction, and in this case it's in pups, since the authors here, they use the model of stress named maternal separation stress, consisting to separate the pups during a period after birth from their mother, and after we can see that the gut effect of this separation can be observed in early life in pups, but also this is a strong imprinting, and we can find these adverse effects even in the at adulthood. At least the maternal separation stress leads to hyperpermeability, as you can see here in the pups, and this was restored by the probiotic treatment, but also not in this study, but in other studies, we can see that the hyperpermeability at adulthood can also be prevented by probiotic treatment. And in this study, it's really interesting because it's one of the rare studies dealing with the absorption function of the intestine, showing that in pups, the maternal separation stress increases the secretion of water and electrolytes in the lumen, and this also can be prevented by the probiotic treatment, showing that, of course, the intestinal barrier function is prevented by the probiotic, but also this gut function, which is the absorption, secretion, water, electrolytes. This is another study coming from our lab in collaboration with the team of Eric Oswald, showing that dealing with the effect, at least a probiotic at adulthood or a probiotic in pups can lead to similar effects somewhat. So, in this study, we have treated adult rats with a probiotic, which is E. coli Nissle, 1917, and this is non-pathogenic E. coli with probiotic effects. And this probiotic, the wild type of this probiotic, was given to the adult rats, and we have treated also the rats with other commensal E. coli able to produce colibactin, which is a substance produced by E. coli Nissle, to see if this probiotic can have a beneficial effect against inflammation, in this case, induced by DSS in the drinking water, if E. coli Nissle can have a beneficial effect, and if this effect is colibactin dependent. As we can see, E. coli Nissle inhibits inflammation, but the other strains, the wild type producing colibactin or the mutant here who does not produce colibactin, have no effect on the inflammation. So this confirms previous results showing the anti-inflammatory possibility of E. coli Nissle. But in this case, we have treated animals, the mother of animals, during the end of the pregnancy, and we have shown that this treatment with E. coli Nissle leads to a colonization of the gut of the neonates, and also this colonization is long-lasting through the life, and even can be transmitted to other generations. Thank you very much. Sorry. So, if you treat the animals with this E. coli Nissle, the mothers, this is transmitted to the neonates and after in the adulthood we have taken these animals who had Nissle in their gut and we induced colitis. And as you can see, when this E. coli Nissle is acquired during from maternal to neonate transmission, the anti-inflammatory effect disappears. And also, in addition, we can see that in early stage of life, we can find an increase of blood translocation of this bacteria, meaning that a risk of bacteremia can be associated with this kind of probiotic administration. Let's move now to another notion of the intestinal barrier function. The epithelial barrier, of course, is the most studied, but mucus barrier is also very important for the intestinal barrier function. And in this study, we have shown that when we submit animals to a stress and we look for the biochemical structure of mucins, we see that the water avoidance stress, which is a stress, subchronic stress, induces an effect on the O-glycosylation of the mucins. And this effect is elongation of lactosaminic chain of these mucins containing O-glycans. And you can see here the appearance of these heavy chains. And when you treat the animals with this probiotic, this kind of effect disappeared. The most important is that these changes in O-glycosylation of mucins is traduced by changes in the structure of mucus. And here, when we make a surface imaging by atomic force microscopy of this mucus isolated from animals, here you can see by this technique a cohesive mucus layer, which is here more flattened and it loses cohesive properties. And this chaotic image is mostly restored by the probiotic treatment, showing that this probiotic and perhaps others, but this is another debate, we have to evaluate one by one, can have an effect on the intestinal epithelial barrier, as I said previously, but also on the mucus barrier, and this is very important for the beneficial effect associated with this probiotic. Let's move now to another area of effect of probiotics, which is visceral sensitivity. And here again, I take the example of Lactobacillus farciminis, which here in vivo, in this time, prevent the hypersensitivity induced by stress. Here you have the control curve, here we have the stress curve, and the red one is the restoration by the probiotic treatment of this alteration in visceral sensitivity, measured as the number of abdominal contractions. And when we take these animals and we look for activation in spinal and supraspinal sites, we have found that, of course, stress and colorectal distension increased the FOS-positive cells in the sacral zone of the spinal cord and in the lumbar also, and this was diminished by the probiotic, and the same thing was happened in the hypothalamus in the paraventriculus nucleus. Looking the properties of this probiotic, which was very well studied in the lab, on the intestinal epithelial barrier, meaning reduction of the stress-induced hyperpermeability and the effect on visceral sensitivity, we have found we tried to find if there is a relation between the reduction of hyperpermeability and hypersensitivity induced by stress. And this is an experience showing clearly in animals at least in rodents, that there is a cause-effect relationship between hyperpermeability induced by stress and visceral hypersensitivity induced by stress. And here, they are stressed animals. Here is the control animals, and in this time we have treated the animals with a pharmacological agent which blocks tight junction opening, and in these animals, treated with this agent, you can see that the permeability is reduced. Here is the hyperpermeability by stress, 0 to 6 hours after, 6 to 44 hours after, and in both time points it's reduced by the pharmacological agent, and of course the visceral sensitivity, as you can see here, is also reduced by this agent, showing the cause-effect relationship between these two phenomena. So a possible way of action by the probiotics is on visceral sensitivity is to prevent the impairment of intestinal epithelial barrier induced by stress. Here is another way to think about how probiotic can act on visceral pain, and it's this elegant work comes from McMaster University, from the Body and Brain Institute, showing that this Lactobacillus reiteri has an effect on the spinal nerve firing, and here is the effect, the reduction of the mesenteric nerve spontaneous firing by this probiotic and his conditioned medium. And the authors have shown that the probiotic or its conditioned medium is also able to reduce the capsaicin-induced spinal excitatory response, and they have shown also that this effect, and they have also shown that this bacteria is also able to reduce the excitatory response induced by distension, so by capsaicin and distension, and when they see the treatment of with an antagonist of TRPV or the knockout animals, they have also shown that this effect is reduced, showing that the antinociceptive effect of this Lactobacillus reiteri is associated with TRPV1 channel action. And this is very interesting because sometimes preclinical studies fits with human studies. This Lactobacillus reiteri is also has an efficacy in infants suffering from colics. Yes. Yes. Okay. So let's move now to the HPX response and behaviors and the role of probiotics in this. Here again, I report the example of Lactobacillus farciminis, who has an effect, as we have seen in intestinal permeability, leading to a reduction of the LPS load in the portal blood, and we have shown also that the animals treated like this, they have 2 hours after stress a reduction in their corticosteroinemia. And when you in the other panel here is the treatment of the animals with another pharmacological agent, which is MLC-key inhibitor. It means this one prevents the tight junction opening. And here again, we can see that we have a decrease of the LPS upload in the stress conditions, and also a decrease of corticosteroinemia. And this study led us to thought that the prevention of gut leakiness through decrease of LPS translocation by a probiotic can attenuate the HPA axis response. This is also some data obtained by the Cork team, showing that another probiotic reduces stress, anxiety, and depression-like behavior, as shown here in the open field test. This is the stressed animals. They spend more time open field, and this is the effect of the probiotic. This is the effect of probiotic, and here is also the effect of probiotic, showing that it exerts some anti-anxiety effect. But the key point in this study is that in vagotomized animals, this effect, beneficial effect of the probiotic, does not occur, clearly showing that the vagus nerve can propagate the positive message. The same team have obtained beneficial effect, antinociceptive effect, with another probiotic, which is Bifidobacterium infantis, and they have shown that this probiotic, and not this one, is able to increase the threshold of colorectal distension in Sprague-Dulé layer rats, but also in this kind of rats which are hypersensitive genetically. And this example is very interesting because the Bifidobacterium infantis, this one, has been also be approved in a clinical trial, showing that this one, B. infantis, and not another one, Salivarius, is able to reduce the composite score in IBS patients when they are treated during with 2 weeks with this probiotic. Yes, finished. Yes. The conclusion is My conclusion will be that there are several ways to act. Of course, restoration of the epithelial barrier and mucous barrier, avoiding the immune activation, and nerve fiber sensitization. But there are now a big field, which, in my opinion, is quite black box, which bacterial metabolite, direct or indirect action, adhesion, mucus interaction, bacterial interactions, in order to provide effects. And there are, every day we have some new mediators which can be involved in the beneficial effect of the probiotics. And I cite just the example of another team in Toulouse, which Nicolas Senac is in the room, and he published very recently today a Nature communication showing that lipidic mediators can also be beneficial and a way of action of probiotics. So we have, it's a lot of things to see about probiotics and to understand their effects. And the most difficult, in my opinion, will be to make an appropriate screening, which pathology and which way to screen, what we have to look for. Thank you for your attention, and sorry for...